Hello, this is going to be Kapp's blog number 50, inaugural show. This is a great show. We have a student in there from other classes, and Emily. So I will be kind of lecturing with my mask. I'm going to hold off a little bit. Let's see if anyone else from the PDA. All right, I'm getting ready to go. I'm going to share my screen. And we're going to present. Hello, everyone. This is Kapp's blog number 50. So I've been doing this 50 times. Wow, quite an area. Today, we're going to be talking a little bit about what we call cell and gene therapy. And so there's a number of discussions, there's a number of groups that are going on, and they're going to be talking about cell and gene therapy in October. And so I really want to promote these talks with us. And also I want to talk a little bit about maybe what cell and gene therapy is. So when you go on these talks, you can be there with some knowledge. Uh-oh. All right. So this is Kapp's blogs with me, Nick Kapp, PhD, your SFPDA SC academic advisor, and the members of the PDA member audience. It's a weekly podcast, roughly about 5 to 10 minutes long. Help us help you. We tend to inform and educate a new generation of pharma manufacturers through a discussion of helpful links, stories, and interviews. Help us help you. In the coming weeks, we are going to try and do a little bit more of this. If you have any ideas for what you would like to hear about, or what you'd like to publish, or if you would like to present yourself, kind of let us know. And so again, as usual, this is the San Francisco Peritoneal Drug Student Chapter Association. And we are members of PDA. So what I want to talk to you about is this month there are two free meetings going on. One is the Fierce Biology, the Biotech and Cell and Gene Therapy, that is on October 19th. They are having a meeting kind of all day long that you can go to, and it's basically a panel. And then the PDA is also, every year we have a meeting on cell and gene therapy. And this is going to be October 14th. And the time here is wrong. It's roughly about 9 till about 10:30 Pacific Standard Time. And so some of you may be thinking, what is cell and gene therapy? Sorry, I have a mask on. I'm speaking in public from the biotechnology labs at Skyline College. But cell and gene therapy has a lot of promise of what's in the future. So gene therapy is described as the medical field that focuses on the genetic modifications of a cell to produce a therapeutic effect or treatment of disease by repairing or reconstructing defective genetic material. And so what we do in gene therapy is, if somebody has a bad gene, you would introduce the good gene into them and it would work. Or you would introduce a gene that would alleviate the problem of the bad genes that they have. And so in gene therapy, you're delivering the genes to an individual, to either their tissues or to their cells. And so the example that we have here is these are adeno-associated viruses. A lot of times you will use a virus because the virus will target the specific cell and will introduce your gene into the cell. And so some of the, we're using some of the adenoviruses. The Sinovac vaccine is an adenovirus. And so there's a number of different adenoviruses. And so what we would do as manufacturers, we would manufacture this virus with the gene that we want to introduce into the tissue that we want to introduce it. And then you would purify this virus and you would give it to somebody, and then that would be introduced. Cell therapy is a little bit different. Also what we call cellular therapy, cell transportation, cytotherapy. Cell therapy is a therapy in which viable cells are injected, grafted, or implanted into a patient in order to affect a medicinal effect. For example, by transplanting T cells capable of fighting cancer cells, we can give them a patient's cell-mediated immunity and this is a type of immunotherapy that we have, or we're grafting stem cells onto regenerate diseased tissue. So an example of this would be the CAR T cells therapy, where we would basically be taking some of your cells out. We'd be training those cells to recognize your cancer that you have, and you inject those cells back into the body. Those cells would grow and be happy and hopefully cure your disease and not harm you. Another example of that would be, again, we take, you have a heart attack, we can inject cardiac stem cells into the heart, and hopefully those cells would grow and those would replace that tissue that was damaged during your heart attack. And so here's an example of here. And so there's cell therapies, a number of different ways to do it. In auto cell therapy, what we can do is we can take out some of your cells, we can modify them, and then make sure that we put those cells back together. What we don't want to have happen is we don't want to introduce any contaminants, bacteria or viruses or anything else. You also don't want to mix it up, right? So if we take Emily, sorry, Emily's here in the audience. If we take Emily's cells, we modify them, we want to give those cells back to Emily. We can't give them to somebody else because that would be kind of a disastrous effect. So one of the problems with this cell therapy that we have is that you have a batch of one. What you would like to do is maybe get some general cells that you could grow up, you can amplify, and then you can have one batch and you can just have these cells on the shelf and give them to somebody. So that the idea with this is maybe we can take embryonic cardiac stem cells. We can amplify these, you can have them in a hospital. Somebody has a heart attack and you can inject these stem cells into their heart so that they can start replacing their heart material right away. Again, you know, as part of the Peritoneal Drug Association, what we're talking about is that how do we deliver these drugs? And cells and gene therapy isn't going to be like taking aspirin, acetaminophen, where you would ingest those particular drugs. They have to be injected into the circulatory system because, again, your stomach would kind of break those materials down. And so what I want to describe to you, the parenteral route of drug administration, basically par meaning beyond and internal meaning intestine. And so what these are is these are drugs that don't go through the intestine. These are drugs that are injected directly into the body, and they go into the body in other ways than through the gastrointestinal tract. And so again, typically we talk about these drugs as being injected substances. And the thing about these injected substances is they have to be very clean, right? So if you have a aspirin and you don't want it to be contaminated, but if it's contaminated, if it has a little bit of some bacteria inside of it, the GI tract is going to take care of that bacteria. However, when something goes directly into the body, if it has pyrogens, if it has other cells inside of it, it can directly affect the body. And so so we are the West Coast chapter, student chapter, West Coast chapter, PDA. How the PDA kind of help is the PDA is a professional organization where others outside of their company try and help you do your job. And so when you work for a big biopharma company, that company really wants you to kind of know your stuff. But sometimes, you know, what if you're in a small company and you don't know this material yet? What if you're doing something kind of a little outside the box? And so what happens with there is PDA exists because people who work in these companies who have the knowledge, you know, they're not going to give you proprietary knowledge. But the idea is that if we have a number of professionals who talk with one another, who say, hey, this is the way that we should amplify these drugs. This is the way that we should prove that there's not other alternate viruses in our drugs. When we get together like that, we help the industry overall. And so again, it's not in a company's interest for you to start another company to make a drug to replace theirs, but it's in the interest of everyone who does parenteral drugs to basically help one another to make sure that we are making the best drugs that we can. And so that's one of the reasons why I like being a part of the PDA, because again, number one, we get to know people in other companies, and that we as a society are going to make these drugs better by being part of a group. So again, when we look at cell and gene therapy, these are materials that are going to be injected into the body. They're going to be much larger than many of the drugs that we have. So like, again, we're talking even much larger. We're talking about a whole virus or a whole cell rather than, again, some of the immunotherapy drugs that we use, which are basically just protein, which are basically antibodies. These particular drugs, the cell and gene therapy, are regulated by the Center for Biologics Evaluation and Research. That's the CBER in the FDA. And a lot of times what I'd like to try and do is, this is what we call a Form 486 letter. So if you have a company and you get inspected by the FDA and you did something that wasn't all that good, you're going to get a letter like this. And one of the great things about the PDA is because we are a group, if your company gets a letter like this, you know, you probably have a friend who's working in another company, especially if they're a PDA member, and you can call them up and say, hey, you know, kind of what's going on here? Or maybe you took a class from PDA or the West Coast chapter of the PDA on talking about this, so you would have something you would be able to talk about this. What I want to say here is this particular was a group, this is the Utah Cord Bank LL Limited Liability Corporation, DPA Utah Cell Bank, okay? They got a letter in which they were given 48 hours to kind of fix this problem. One of the problems that they have, typical that we have, is failure to establish laboratory controls that include scientifically sound and appropriate specifications, standards, sampling plans, test procedures designated to assure that components, drug product, containers, closures, in-process materials, labeling, and drug products conform to the appropriate standards of identity, strength, quality, and purity. And this is the 21 CFR 211.160B. The federal government doesn't make that, we do. As part of PDA, as part of a group, we all get together and we set those standards for the sampling plans, for the testing procedures, and kind of everything else. And so that's one of the things that the PDA does. And so even though these are new drugs, we have new viruses, new cells that are going around it. We are getting together in October 14th and October 19th, and we're going to have a panel and we're going to talk about these things. And those are going to be our standards. So it's really important that we look at this. And so panels and trainings are meant to help us understand this relatively new field. So we understand that. So I know a lot of you are going to be younger in your field and you may not have a lot of questions. What can you ask this panel? You know, again, you may want to ask the panel, what training is required to manufacture cells? You may want to ask a panel, what is the future of cell and gene therapy? And again, these are some, you know, we're developing the cell and gene therapy to treat diseases that in the past we had no basically cure for. So what are we going to do? We're going to look at it. So what talks do I want you to attend? The Fierce Biology, the Biotech Cell and Gene Therapy is October 19th. The PDA Cell and Gene Therapy occurs every year in October. That's going to be October 14th. Cell and Gene, which is a group, they have a podcast, the Cell and Gene podcast that you can look up. Thermo Fisher has a Cell and Gene Therapy Center, and they would love to sell you all the equipment on that. Some of the trainings that you can go to in terms of basic training, gene therapy by Bluebird Bio, actually for about $400, you can take this class. Stanford has a class called Principles and Practices of Gene Therapy. And then Introduction to Cell and Gene Therapies by Bioprocess International. So these are all classes that you can take for a couple $100. You can go ahead and take that class. So it's a new field out there, cell and gene therapy. And so again, there's a political aspect to this. There's a scientific aspect to this. Everything that we look at has this. So thank you for tuning in and learning a little bit more about cell and gene therapy. This is Kappblog. See you later. Any questions? Hey, Maria, you made it. You are, Maria's in, she's doing her stuff. Oh, Catherine's here, cool. Well, genetic diseases. So if you're missing a certain gene, they can do a gene therapy in order to kind of give you that. So that would be something that would be there. Maybe something like HIV. And so there's certain genes that would give you resistance or better resistance to HIV. That there's a hope of kind of using that for people. You know, a lot of it is kind of editing. You know, so and so can we edit the genome or can we edit the cells? You know, in that person. I think a lot more people are a lot more open to editing somatic cells than they are to the germline. Yeah, you're talking almost like $1 million a treatment. And that is expensive. And hopefully as we do this, the price will go down on this a little bit. But on some of these diseases, you look, if you have to do a transplant or if somebody's in the ER for a month, you're talking a lot of money there. But, again, it's kind of one of those things that, thousands of people need this particular therapy. It's going to be way too expensive in order to do it. What they think about, I think for a diabetic, it's about $100,000 a year to treat a diabetic. And so that's kind of what they think. So for $1 million, you got to give somebody